So it's my pleasure to introduce Dr. Iris Segura-Garcia from the FAU Harbor Branch Oceanographic Institute, and we're excited to hear her talk today about seagrass. Thank you so much. Of course.
Well, good morning, everyone. I already say good morning to all you, mostly my friends. Thank you very much for coming this morning. And today I'm going to give you a little bit brief overview of 2 projects that I am conducting at the moment at Harbor Branch. That they all have one common thing in common, that is the seagrass. And to start my talk, I'm just going to clarify some confusion that sometimes come often in talks. Seagrasses are not seaweeds; they are plants. Seaweeds are more simpler organisms. They just have a whole pass that they tame themselves to the bottom, and they transport nutrients by diffusion into the blades, while seagrasses are 2 vascular plants. They, as any of the terrestrial plants we have in our gardens, they are actually more closely related to lilies, our plants lilies. They are really true plants that they came back to the marine environment. And another thing for these 2 vascular plants, they produce flowers and seeds. And they also have a root system and a rhizome. The rhizome is this stem that grows horizontally, that then helps it to absorb and store nutrients, or also to hold the sediments, to hold into the sediments. And this is one of the first ecological services or importance of seagrasses in the marine industry, holding or stabilizing the nutrients. Other ecological importance or services that seagrasses provide, they are foundation species. That means they provide habitat, but not only habitat, they provide food for many organisms, such as invertebrates, crabs, shrimps, you name it, fishes of different sizes. They are nurseries for several fishes that reproduce offshore, but they are also food for turtles, birds, manatees. We know very well that manatees are voracious herbivores, and also to dolphins. Unfortunately, the seagrasses are endangered. They are threatening coastal degradation, environmental degradation. And this is illustration that I take from Dr. Lapointe here, present with us in the audience, a paper about the enrichment of the lagoon. So these are the good days of the lagoon. We have clear waters, and you see the waters for the sunlight can penetrate to the bottom. The seagrasses are any plant who need the sunlight, right, to grow and to feed themselves through photosynthesis, produce their food, and grow very happily. So in good days, when we have clear waters, the sun can penetrate, and algae and seagrass can grow. There is also, you can see here, there is not too much coastal development. We have a good forest along the coast. The mangroves and other trees that they grow along the coast are if you want, act as a kidney, like filtering nutrients before this water reach the estuary waters. However, the truth these days is very different. We have too much nutrients in the water now. So we can see there's more development, less trees, less mangroves along the coastline. That means there is more input of nutrients into the lagoon. The algae, the microscopic algae, which is called generally as a group phytoplankton, they overgrow because they overcompete the vegetation in the bottom. They are closer to the surface that they can have this light and use this source before, so they can overgrow and dominate the system, right? So now the lagoon, instead of being a seagrass-dominated system, now it's a phytoplankton-dominated system. And we've seen our waters, right? There is very greenish, turbid waters. So we don't have clear waters. That means there is light attenuation, which is one of the main limitations for seagrass growth. And in worse conditions, in worse conditions, this phytoplankton can be developed hard from algal blooms, like overgrow, like in massive densities, that and they can result in fish kills. not only because they can be toxic, but because simply there is suffocating, there is not enough oxygen for fish to live, and they are massive fish kills, but also if these hard-to-man lagoons like for prolonged times, they basically just kill there's no light and they kill the seagrass, and resulting in massive die-offs of seagrass. And I'm going to welcome you to read this paper on nutrient overreachment from Dr. Lagoon. It's very interesting, and it describes this in very, very detail, more than in this morning.
What is the KD ratio?
Sorry?
What is the KD you have there, and what does that stand for?
The what?
KD.
I mean, it's the light decay, the light attenuation. So you see in the previous one, we have pretty much close to 0, and in this one that it's very murky, we have 1.5. So light doesn't penetrate into the bottom. Good question. So this is a good illustration, but I want you to see it with your bare eyes. This is footage that is taken from with an ROV from our pontoon boat, the Discovery pontoon boat, during the boat tours at Harbor Ranch. So if you want to check it out from yourselves, we have boat tours there. Sam is going to tell you more about it at the end. So you see these are moderate turbid water. So now you can see the glare of the sun is still there. It's still not optimal. There's some turbidity. We have epiphytes in the blades. So they are also a result of a heavy load nutrients. They are not good for seagrasses because they are covering the blades for conducting photosynthesis. So it's not optimal, but it's not bad to have a good metal. You can see there's very dense manatee grass right here. For other days, we have very turbid water. You can even see the bottom. And this is most of my days, something in the water. I really have to touch the bottom with my head to be able to see what is in the bottom. And it's nothing there most of the time. And what you will see is that there is basically very bottom. There's just a few shoots here and there. So also the die-off of the seagrasses means that there is nothing holding the sediments, right? So there is also like worsening the situation, right? There is sediments in suspension in the water, so there is even less light coming into that. So this is the same site, believe it or not, just 3 months different. So that is a lot of cycling and periods of good and bad, right? And it depends on how the estuary is, how these changes occur in the estuary. And estuaries, like naturally, they are normally very changing environments. You know, we have input of water, fresh water, but also oceanic water. The depths are different, so there is more evaporation of water. Anyway, they're naturally very changing environments, so they are very susceptible to these changes. So just to reflect these changes, this is a graph for since 1982 to 2024. This is data that was collected by the San Jones River Water Management District, and they are always very supportive and you can share the data. So this is an illustration of how those changes on seagrass we have. Well, the points that I want to point out this morning is when we have massive die-offs of seagrasses. And these arrows indicate not only the die-off of seagrasses, but they are also coincide with we have like super blooms or platon blooms. So it's not a coincidence. This is an occasion. This is the result of this phytoplankton blooms. And just in green, this is the last years, and it seems to be a little bit recovery. It's not optimal, it's not going to historic limits, but we've seen a little bit of recovery. So in total, by 2022 or 2021, I remember, we have reported seagrass die-off of 95% of our seagrasses in the entire level. So this is terrible news, right? So that means that the ecological services that I mentioned to you before are no longer provided. We don't have food, we don't have habitat for most of our key actors in our food webs. So this has alerted not only us scientists, but the community and managers of the estuary to increase the efforts of restoring our marine ecosystems. And with that, I mean like developing new nurseries and for outplanting seagrass plants to facilitate the recovery of this important foundation ecosystem. And this is taking advantage of the asexual growth of seagrasses. As I mentioned before, like they can grow with this rhizome. So if it's like our lungs, you buy a piece of soil, you put it and it starts growing. If you have good conditions to water, you have good light, it will grow, right? But the problem with this practice is that we don't know. We might be planting the same plant over and over and over, and just growing a clonal meadow. That means that everybody is identical, so they might, they just have exactly the same information. So, and that could lead to very low genetic diversity in these meadows. And below that, with no genetic diversity, we cannot evolve, right? Like everybody will have the same information and same characteristics and not be able to respond to disturbance. So with these cartoons or figures, I wanted to illustrate like how important is genetic diversity to evolve. In the population A of seagrass and of seagrasses, we have different colors, and that is a representation of genetic diversity. There's a high genetic diversity, we have different colors. In the population B, we have pretty much all of them are the same color, so that is a low genetic seagrass meadow if you want. So in the middle, we have a disturbance, in this case, the stressors that we are aiming to test. And if you have some genetic diversity to respond to that, you might be able to survive and to adapt to different conditions. While in population that you don't have like limited genetic diversity, most of your individuals will die. There is no divergence, and that can lead you to local extinction. I forgot to mention that there is this. This is the one study that I am referencing here. There has been suggested that higher meadows with higher genetic diversity can be more resistant to a stressor, and this has been tested in only 1 of our 7 seagrasses that occur here in the lagoon. So there are many unanswered questions, right? In order to have a successful seagrass restoration. With a diet of 95%, we don't know what is the status of our wild population or whatever has survived in the lagoon or actually along Florida. So we don't know what is the extent of genetic diversity or how they are differentiated. So we are starting to, so the seagrass didn't die all at once like a mat, so they were patches that were more resistant to others. So what is surviving? What does this patchiness mean for the sexual reproduction in the seagrass, right? So also if this, for the patches that survive, what is this genetic function, functional variants are making them to survive? On if the worst case scenario, how much genetic information, important genetic information for evolution have been wiped out with these diodes. So in order to answer all these questions, we have a project an ongoing project, and this assessment of the genomic variability, and you will see genetic genomic like exchange sometimes, but genomic means at a genome level, not only a few genes, but at the whole genome level. In order to the aim of this study is to assess like what is the genetic variability that can be that can describe like some functional resistance to this stressor that they are facing these days. And this is the project that I am conducting with as part of the Seagrass Restoration Technology Development Initiative that is led by Maud in laboratory, and with my collaborators Michael McCoy from Harbor Branch and Kristen McCoy, previously Harbor Branch, now at Florida Oceanographic Society. So as I mentioned before, most of the studies and most of the restoration practices today, and the few studies that we have on genetic variability are focused on shoal grass. This is the dominated species in the IRL. But the second most abundant species is the manatee seagrass. It's very common in all South Florida, but it's very important for in the river lagoon. And also we promote more diverse restoration, not only focus on one species, also diversity at genetic level, but also at the species level can provide more resilience to the system, right? Also, manatee seagrass have some life features that are very important for restoration, very attractive for restoration. Their rhizome elongates, grows very, very, very fast. Also, they can perform well as colonizers. and as persistent species. So these characteristics are very attractive to start increasing the efforts on restoring and growing them in nurseries and restoring place using these species. But as I said, genetic diversity is very important, right? To know before we start restoring wild sites or growing seagrasses in nurseries. But we don't know much about genetic diversity in Florida. The only study of manatee seagrass genetic study was conducted in the Florida Keys and Tampa Bay, and there was a comparison with the Bahamas and Bermuda. So we still don't know what is happening in the rest of Florida. Lucky me, right? Now I can do this study. So as I mentioned before, I'm using a whole genome approach, learning a comprehensive genetic variability of the whole species. So just as a comparison, and that's what I have my pipeline for workflow, is that I am working with more than 10,000 markers or genes, if you want to call it, right? So that's very, very high resolution. That technology, the genetic technology these days, they provide you a high resolution and it's very cost effective, right? The same money that I will spend looking at 10 genes, I will probably have this pretty much the same as these thousands of genes for not much more money. So it's way high resolution, really high cost effective. So one of the advantages of this approach is that I can distinguish functional genetic diversity, right? And with that, we really want to include these because we want to identify genotypes that can be adapted for different conditions. And these will inform the nurseries, right? The land nurseries are very space limited. So if I can identify these genotypes that I can grow in my zone because I live in Martin County, so I know that I have higher salinity or lower salinity than we have. And anyway, different specific local characteristics, so I can start prioritizing the growing of plants that are resistant to my particular conditions for my restoration sites. And the idea is to have an evolutionary informed seagrass restoration, meaning that I will just plant the adapted genotypes to these local conditions. This is a long term, so I'm just learning about what is the genetic diversity of the wild populations. So far, I have sampled only along the Atlantic coast of Florida, and this is one of my first results of the population genomics. That means I'm comparing all the populations and see like how different they are. And amongst all my thousands of loci that I told you I'm analyzing, this is like a condensation of all this variability into axes, right? So I can have thousands of axes, and so we condensate it all into axes. And the H dot here represents a plant sample, and the closer these samples are the more genetic similar they are, right? So the color code is from Biscayne Bay, Florida Keys, the Central IRL, Northern IRL, Southern IRL, and Lake Worth. So we can see, like with our graph that what we see at first sight is like the Miami and Florida Keys form a group. They're more closely related. The Southern IRL and Lake Worth also form a cluster, and the Central IRL and the Northern IRL also form a cluster, but they seem to be very geographically coherent, right? Another level of genetic differentiation that I'm seeing or I'm looking at is the level of clonality. And this y-axis is the genetic distance, meaning that the more distant they are, the less related you are, right? And this line, the dashed line, is the threshold for you can be called a clone, like identical clone. And I can draw this line here because I have technical replicates. That means that I sample the same plant or I genotype the same plant different times, duplicates or triplicates. So I know that this plant has to have exactly the same genetic information. So that is my threshold. From this line below, everybody is a clone, and below, above the line, we have plants that are a product of reproduction, the sexual reproduction. Means that there is a flower they fertilize and they produce a seed, and there is a plant growing. So I have identified 11 clones. We have more clones in the central IRL and Oxahatchee. We have several clones also in Biscayne Bay, but also several plants that were product of sexual reproduction. Another important thing, I don't really see it very well in this figure, but hopefully you can see it. I mean, the color codes are similar to the previous figure, where I have also samples from nurseries, from the Florida Oceanographic Nursery and the Harbor Branch Nursery, and we can see this is very important for us, and it's a very good result now, because now they are conducting restoration like these years, right? I mean, these years when they're have some plant. So this will inform them which plants can be planted and where, at least they know the source of their plants, or which plants are different, so they can start growing more and more informed growing right in their nurseries. So this is a very good result so far, and this is just like I'm just starting to start then. I just started so these I just received my data like no more than a week ago. But this is what I have so far. So my initial findings indicate that there's complex patterns of genomic diversity. There's a higher clonality in the central and the River Lagoon compared to the South and Biscayne Bay. We have confirmed or identified some plant sources that are growing in local nurseries. And these, as I mentioned before, they can inform the potential suitable restoration sites for these plantings. And hopefully with that, we can increase the success of seagrass restoration, and with that, provide more habitat for our coastal fishes, or restaurant fishes that depend on these foundational species. And one of the species that I am more interested, or I've been interested for years now, and studying it, is the spotted sea trout. This is an estuary-independent species, and you don't know, it distributes from Massachusetts to the Bay of Campeche in Mexico, where they eat a lot, and they call it actually corvina. So if you go to a restaurant there, they normally have like a ceviche, ceviche de corvina. So these, one of the characteristics that interests me for this species is like, unlike other fishes in the same family, the drum family, that reproduce offshore, these species reproduce in the estuaries. Their whole life cycle depends on the estuary, and a lot in the seagrass beds. So just a briefly explanation of the cycle, description of the cycle. In the spring, summer, the males start producing a drumming sound. That's what they are called, drums, and they attract the females. Then the female release the eggs, and they are fertilized. And 18 hours later, the larvae go to the bottom, which is very unusual for fishes. They normally go to the surface. And then like days later, they start developing a small mouth, and then they can start feeding on zooplankton, copepods, and then as they grow, juveniles, and they repeat the cycle. But all this happens within the same estuary where they are born. They are known as non-migratory species. They don't exchange like they don't go out in shore and when the coastal areas and change environment, typically, right? So that means like if you're gonna live in the same estuary, in the same environmental conditions, you better adapt to them, right? But these changes are happening very rapidly. So are they adapting, or what's happening with this species, right? Where this species has been, it's been affected by overfishing back in the days. There was a very important commercial fisheries that collapsed in the 70s because there was the proper regulations allow them to catch small fishes, so they don't allow to reproduce, and the fishery collapsed. So these nowadays they only sustain a recreational fishery and only moderate commercial fishery. Therefore, they have a high economic value, but they have, of course, their ecological value, right? They are predatory species, so that means they are a major player in the in the food webs in the estuary, and also they are prey of some top predators, not only us, because we love to eat sea trap, but also our resident bottlenose dolphins. So these species is very well regulated. They have substantial fishing regulations in place. In Florida, they are managed in 5 different management zones, and is the only state among the distribution of the species that is managed in more than one management zone. So without, that tells you a lot about what is the differences or how distinct these estuaries are, but not only the estuaries, the environment, but the way the population is responding, the dynamics of the population in each estuary is very different. That's why we have different backsides with different closing seasons and so forth, right? That they're very different in between these zones. So with these very refined fishery regulation, we expect that the fishery will recover, right? Well, what we're seeing is the opposite. So this is some plots from the last citral stock assessment. In the y-axis, we have the spawning biomass in metric tons. Spawning biomass is like how many fishes can be reproduced in that year, right? So we still continue to see a decline. You see decline, but the point that I want to drag your attention is the point in 2017. In all the 4 management zones, and that is back in the day, they used to have 4 management zones. Today we have 5. It's like even more refined. So the regulations are very dynamic. They are changing as they see the response of the population. So the IRL is in this zone, but in 2017 is when we have seen the lowest spawning biomass in 60 years. So this decline is still ongoing. So if the fishing is controlled, there is not much mortality for fishing. There has to be the environment that is killing our fishing. So as I mentioned before, we have a phytoplankton-dominated environment, fish kills, input of fresh water, the seagrass is gone, and with these fish kills that means like high mortality of fishes, we are not only wiping out individuals out of the system, we are also wiping out important genetic diversity that will helping these species to adapt to the ecosystem or also to rebound to the story or to more sustainable levels. So again, to learn more about the genetic diversity of these, it's really important to learn about the genetic variability of these species in order to inform protection and management action, right? So I use again the similar genomic approach. I'm using thousands of markers. I am going to show you results only about the Atlantic coast of Florida, including the Jacksonville area, and the IRL all the way to Jensen. I don't have samples for the rest, which would be very interesting to compare, but I have them. So I'm sending you this approach a lot. That this is like the one and only. That's very high resolution, and this is just an example of how this approach compares to the traditional methods that we used before. So this is a plot that tells you the ancestry of all the fishes, each varies in the fish. But the only thing that I wanted to show you today, for simplicity, is there are 2 populations, right? And they are color coded as in the management zone, according to the management zone. So in green we have the northeast, in blues we have the central east cluster or management zone. So we have 2 populations highly differentiated. What we used to know before, using only 21 markers, is like these 2 populations for 1. So that we are with using this method, we increase like very significantly the resolution of these genetic assessments. So another beauty of this approach is that I can distinguish between neutral markers, meaning that they are not affected by evolution, and diversifying markers that they will give me information about how much they can adapt, or what are the potential that they have to adapt to different environments. I don't want to go into too much detail on that, but I'm going to show you the results of that, of these, of the analysis of these markers. So again, my plot of genetic variability into axes. We have the green individuals from the northeast clearly separated from the central east, including all the regions of the Indian River Lagoon that I sampled. So they are just different shades of blue. There are two individuals that were sampled in Denison Beach that they clustered to the northeast Jacksonville area. So I'm telling you that it's a non-migratory species, right? So I'm investigating how these guys ended up here in the Indian River. So I'm gonna dig more into that, but there is some. What I'm showing you here is there is some directional migration from the northeast into the south IRL. And just briefly, like this number here, I mean, it's an index of differentiation. When it's close to 0, it's not differentiated. When it's close to 1, it's highly differentiated. I told you that I can separate between some functional and non-functional genes, if you want to say it for simplicity. So when I use the neutral genes, we have close to 0. When we have the adaptive markers, there is 1 order of magnitude more different, meaning that adaptation has to do a lot with this differentiation. Let me just go back a little bit. So I don't know if I want to see, but I think there is some genetic differentiation of my population within the Indian River Lagoon, which might not be crazy because I mean there is a lot of different habitats along the Indian River Lagoon. It's a very long estuary that and also they have different climates and different habitat characteristics. So to look at a finer scale, I only run the analysis only for the individuals from the Indian River Lagoon, and I think these guys are separated. I mean, the index is very close to 0, but and still there is a gap that is differentiating the northern IRL and Banana River to the southern IRL, and the same with the diversifying loci. It's 1 order of magnitude higher, but it's not tremendously high. But I think this is what I'm learning so far. You know that there is some fine scale genetic differentiation between the 2 management zones, so they are well managed based on genetic differentiation. There is some directional contemporary gene flow between the 2 estuaries. Adaptation seems to be a stronger driver of differentiation between the estuaries and potentially between estuaries. And so this within estuary differentiation might reflect adaptation to different spawning environments. Sea trout are known to adapt or not to adapt, to perform better in their spawning environments. Salinity is a crucial factor, environmental factor for their survival and retention of the eggs. So they seem to perform better in salinities where they respond. So that might be a part of the adaptation that I'm seeing in genetic differentiation, or to be also due to habitat fractionation. Like I was just telling you, like this, but actually probably both, because we see the differentiation in both markers, right? So there is probably patches of seagrasses where they are just like simply there are no connection in between, so they are spawning in different habitats. So hopefully with this information we can inform the management and regulations that habitat is very important for this species. It's not only limiting the fishing mortality is important, but also restoring or protecting their environment is very important. So that could probably be an integrative management, like protecting the environment and the species, and hopefully recover the extreme coastal fish populations. And this is very important because, as mentioned before, there are top predators that depend on these coastal fisheries. And this study is on to learn about how the changes in foundation species, the seagrasses, are affecting the diets of our resident bottlenose dolphins here in the Indian River Lagoon. And this is a project that I'm conducting in collaboration with Seagrass World Research Institute and the same collaborators Krista and my colleague in Harbor Branch and Florida Oceanographic. So knowing in detail the diets of predators is crucial to learn about their ecology and evolution. What is their role as predators in the food webs? Learn about their prey selection, which I suppose is very important when we talk about species that are important for fisheries. If there is competition with these top predators and fish populations. Also to learn more about their foraging strategies and resource specializations. For the focus of this study is what happens when we don't have seagrass. We don't have seagrass. We know the bottlenose dolphin in Florida, their diet is only focused on the fish. They eat only fish. They have preferred prey items. Sonorous fishes are the sound-producing fishes are the preferred prey because they use or they seem to indicate that they use passive listening to detect their prey. So here in this list we have 5 drums. So the sound trout, the black drum, the red drum, and the sea trout at the bottom. But all of these are their preferred fish. That means that even if they are in low quantities, in low abundance, they will still prefer and go for these fish. Well, they can also feed on non-preferred fish, so they are highly available. Amino mullets are everywhere, finfish, and so they are highly available, and they go for them. The common thing amongst all these species is that all these species are seagrass associated, so meaning that the seagrass are indeed important foraging habitat for bottlenose dolphins. So what happened now that we don't have seagrass beds? We lost the critical habitat of all these prey items for dolphins. So to learn more about what dolphins are eating in the last decade, we examined using just visual examination, but also molecular tools, stomachs that were collected by Hopsy World Research Institute, opportunistically during strandings that animals were dead or they died. So I went into these bags of digested fish that was very recent. If you can, this is just examples of some of the stomachs. You can see like several prey items partially digested. But one thing I want to show here is like there is the body condition of the animals, right? So even though here, this one is a good animal, so you see there are several prey, but yeah, they're eating well. There may be other cause of death, but this female was emaciated. This guy is thin. Nonetheless, they have a lot of fish in their gut. These animals seem to be eating very well. Nonetheless, it's emaciated. So why is that? So yeah, with the use of molecular tools, I have been able to identify these prey items. We still see the preferred fish, goldfish, gray snappers, mullets, a lot of mullets, sheepheads, quite a few of them. Or surprisingly, we see pipefish. We might have pipefish feeding at bottlenose dolphin. We also have American shad, which is a freshwater species, and they occur in the San John's River and the San Miguel's River. So meaning that dolphins are encouraging to these rivers, freshwater rivers, to find food, which is not good for dolphins. Dolphins don't like fresh water. They have skin lesions when they enter into fresh water, so that's not good for them. We also find American eel, bunch-tailed gophers that have been listed before in Sarasota, sea breams, and tilapia, blue tilapia, which is invasive species. So what we are learning from this study is like dolphins are preying in lower trophic levels, and they're likely with lower caloric and energy content. This is why they are thin or emaciated. They are preying also in more cryptic species, like imagine a dolphin chasing a pipefish. So, and there's more energy demand to capture and handling their food. So this increase of energetic cost for foraging compared to just listening to your food and going for them is also like resulting in poor body condition. And this is proved because 76% of the cases that we examined were emaciated. So the loss of seagrasses are leading to starvation or emaciation in bottlenose dolphin. So yes, the changes in environment are affecting our dolphin population extremely. So with these three studies, I hope I have conveyed this morning that seagrasses are the backbone of the Indian River Lagoon. Without seagrasses, the food web is not complete and it's affecting us all, not only as fishers but also as community. If we don't have fresh water, clean waters, we don't have good health, we don't have healthy fish to eat, people won't visit us, we won't have the economy and the good life that we all like to enjoy in this estuary, living along this estuary. And lastly, I just want to thank all the people that have helped me collecting samples data for these three different projects, the funding and sources, and Marine Laboratory for the Seagrass project, the Dolphin and the Sea Truck project have been funded with the license plate of the Harbor Branch Oceanographic Foundation, and all the institutions that are part of these studies. And finally, let's start. Thank you for listening.
Could you comment on the impact of the water releases from the important on the spot? I keep reading about it in the local newspaper. So, especially the impact on the seagrass.
Yeah, well, it's freshwater input, right, into the lagoon, and most times, Dr. Lapomo, let me lie here. They're carrying a lot of nutrients into the lagoon, so that's not good news for our lagoon. They seem to be more restricted, but I really can't say much because that's like a bit political or like more administration, like how they are managing the releases. So no, nobody likes them. But this water have to come out somehow out of the lake O. And the thing is, the main problem is that the natural water flow of the lake O into the Everglades has been restricted or changed for many years, and they instead decided to create canals into the lagoon as the release for that, and that was not foreseen to be a problem at that point, but it is, right? So to revert this damage or to revert this practice of the years, it takes everybody, right, from all levels. So I guess I cannot say too much about that.
It's bad,
It's bad, yeah.
It's bad.
Yes.
So I understood you said that within the IRL clonal production of seagrass, that's the dominant form of reproduction?
That's the dominant thing.
Yeah.
So that's historically the seagrasses are being thought to be dominantly clonal, right? Because that's how they grow, the horizontal rhizome. But with the new genetic techniques, we know that there is more genetic diversity than was originally thought. And
I'm guessing, I'm guessing there's no idea to assess gene flow or how.
oh yeah, I'm doing it. Yeah.
With so you can tell how gene exchange in my farm.
Yeah. Definitely. And I think this is where we choose this very high resolution approach to have no doubts, right? Like it's not like comparing 8 genes, like part of the chance that 8 genes are similar. We're very limited. So we are like looking at the big picture, right? Like if I told you a clone, it's right. That it is.
I think any success stories around the restoration of seagrass around
Restoration. There are a slightly success this year. We have seen more grass than we used to be in the Banana River, but it's a more protected zone, and in the north of the IRL system, we have almost, I don't want to lie, but I think it's not 100%, close to 100% seagrass coverage of that. So that means that the environment is resilient, right? If you let it be, if you do have good water quality, the seagrass can come back. No, there is seeds there. Remember, there's this sexual reproduction impact that it's released, and when the conditions are good, they may just sprout and just like flourish, right? So that is a good news for the northern part of the IRL. Good news because more funding for research and restoration. Restoration for marine environments is still like kind of a new thing. I mean, while we have restored forests for many years, marine ecosystem restoration is very like undeveloped, right? So we now with the coral disease, we are learning more about coral restoration. We are also in the West Coast, we are restoring forest, hill forest. So for us here in the Indian River Lagoon, every year is a learning. It's a learning. So we are learning. So now we know that we probably need to gauge the restoration size to avoid herbivory, at least while the seagrass are growing their roots and they really anchor to the sediments, right? So we are learning. So I think that that's good. That's good news that we now know that probably planting other seagrass is also might be good, only not focusing on one species that is a colonizer might not the only answer. So I think these are good news that we are learning, and the seagrass restoration is just starting. It's, I think the first projects were started in 2018. So this year, there is a lot of restoration ongoing, and the good thing is that now everybody's on the same page. They know that genetic diversity is important. So we are all like collaborating. There is a lot of partnership to increase the success of restoration in that.
The tilapia are invasive.
Yes.
So are they out competing native fishes?
Yeah, I mean, at least some of the tilapia, they like for the largemouth bass and smallmouth bass, they like to make sand pit nests so that the tilapia, guess who's more aggressive? So I would say over time, yeah, the tilapia are getting the better nesting sites. Over time, they are becoming more successful in now competing with species.
Do dolphins and sharks like to eat them?
But according to the graph there, yeah, at least one species of tilapia. I don't know about sharks too much, but the dolphins apparently are eating them now. And we've seen some species collected right at the inlet, so even though they're considered freshwater fish, are salt tolerant. Probably do less well than saltwater, but still survive.
The seagrass restoration, all that work with the oyster and mollusk restoration that seems to be going on around that, at least in the St. Lucie side and the store and all that.
So how did restoration versus to oyster?
Seagrass. I mean, you see a lot of mollusks and oysters and all that being put out in the near Stewart and St. Lucie River as well.
So this is also part of restoring the living shores, right? Like it's like these are our best friends, right? Like everything that is filtering and removing nutrients from the water are very welcome. So there is like a lot of efforts to restore also clams and oysters that are the natural filter in the system. There are also stories of success and much. I think everybody is in some page. Like now we have to synchronically like restore, keep restoration of oyster reefs, and at the same time that we are restoring reefs. I mean, everything goes back to water quality, right? We have clear waters. We don't have the heavy nutrient input. Our ecosystem can go back to that, but it's a big effort of everybody, starting from us to all levels of administration.
Yeah, question. I've done gardening all my life. I've never grown seagrass. So I'm curious, how do you grow seagrass in nurseries? Do you do it from seed or how do you do it?
From plants. So one of the common practice now in the nurseries is like the pieces of the seagrass plant will wash into the shore. So they pick them up. Hopefully they are viable to grow, so they just plant them in sand trays in their nurseries, the nurseries, and they use that for replanting. Yeah, it's very simple. I mean, we are taking advantage of that asexual reproduction. They just got a piece, and then just put them in the sand and
What's the salinity of the water? So is it salt water or is it?
Yes,
Yeah.
yeah, it's saltwater, they're all over the marine. There are grasses in freshwater too, but in our story, there are 7 species that they have preferences, like desalinated and others, but for the most part, they are all marine marine water.
Does Harbor Branch have some places where one can see this?
Yes, we do. Yeah, there's just like, as we speak, they are probably building a greenhouse. So we have a big expansion of the Harbor Branch Seagrass nursery. So that's able to, it's up for visit too.
Is that part of
gonna
the tour?
It's part of it. I'm not gonna talk about that. Sam's gonna talk about it. Sam can inform you about that. We have Sam with us this morning, which is part of the education team and outreach in Harbor Branch, so they can tell you about the boat tours and what that includes and also some other lectures that we have in the campus. More questions about the seagrass or genetics?
Does seagrass grow in the bucket?
No, I'm there. No, they don't.
Right, yeah.
Yeah, I mean, they definitely like better sediments to grow. This is a great problem, no? And dredging muck to mitigate this and to clean the substrate to plant it's a millionaire investment, right? To do that. So what we have to do is stop creating muck. I mean, stop like providing the source for this muck to produce, right? So I mean there might be some actions later to remove the muck, but that's not an easy task to do. So instead of creating more contamination, we have to prevent this to happen. Right? Thank you.
Just a quick question, having been from Maryland, I'm more familiar with the Chesapeake.
Yeah.
I feel like they've been doing seagrass restoration for quite a while. Are any of those models or techniques useful or applicable to the IRL?
I will say yes, and it's definitely that's part of the models that we are following. They've been very successful restoring the submerged vegetation, and they're very good success. And we are also learning from each other, like all the institutions that are involved in restoration, I think they are collaborating. So they are always knowledge sharing, like this worked for me, this didn't work for me, I'm gonna try this, you should try that. So there's a lot of knowledge sharing, and definitely we are learning from these success stories, like they just big. Lisa?
Oh, I was just curious how far on average the pollen travels from seagrass.
I don't know the exact distance of that. I know that it can travel in that, but there are also some crustaceans, worms that can transport the pollen from plants. I have it. I want to show it to them. I have some photos. I was not prepared. Here is what I'm trying to these are my seeds. These are some pollinators. This is from a picture from a friend and colleague in the Caribbean. So these are grain pollens and pollen grains. And there is a video attached to this paper also that they show that at night how this amphipods, mainly crustaceans, that they move around the flowers and they can transport it to different. So they have pollinators, just as terrestrial plants, right? Anything else?